Hello, my name is Cassie Wood, and I am a fourth year biomedical sciences student in Dr. Jonathan Canton's lab. This video will focus on my research over the past year, which focuses on characterization of APOL7C in macromolecule escape from phagosomes in dendritic cells. Now, that title contains a fair amount of jargon, so let's break it down. To understand my project, you'll need a brief primer on dendritic cells. Specifically, conventional type 1 dendritic cells are CDC1s, which are a type of immune cell that will surveil the body for signs of danger, such as dead cell debris from tumor cells, and are also involved in mounting a robust adaptive immune response through the process of antigen cross-presentation. But what is cross-presentation? Well, cross-presentation is the process by which dendritic cells will internalize extracellular protein into membrane-bound organelles called phagosomes. Now, typically, this internalized material will be delivered to lysosomes for degradation. However, in some cases, in the case of cross-presentation, this material escapes directly from the phagosome to the cytosol, where it is then processed, loaded onto MHC1, and presented at the cell surface where they can prime naive CD8 T cells and help mount a robust adaptive immune response, which is critical for eliminating infected or cancerous cells. However, a knowledge gap exists at this step of cross-presentation, the leakage step, and that will be the focus of my project. Upon joining the lab, they had just completed some important preliminary work which identified APOL7C as a potential protein involved in this process. APOL7C is uniquely expressed in dendritic cells, which are our key cross-presenting cells. It is upregulated the most out of any in the APOL family, and APOL7C contains an ancient colicin-like pore-forming domain. Together, this suggests that APOL7C may have a role in macromolecule transfer from the phagosome to the cytosol. That brings me to my hypothesis that APOL7C is recruited to phagosomes, where it can then trigger the leakage of cytosolic protein. So, of all the data I'm about to show, I had a primary role in executing, imaging, and collecting the data. We primarily used immunofluorescent assays and confocal microscopy. So let's jump right into my first objective, which is APOL7C recruited to phagosomes, and if so, when? For this, we fed our dendritic cells ova-coated beads, shown in green, and these were in cells that had APOL7C engineered to be fused to a fluorescent protein M cherry. We see distinct magenta rings around ova beads, indicating the presence of APOL7C at the phagosome. We quantify this in figure B as being a late event because we see most APOL7C recruitment at 2 to 3 and sometimes even 4 hours. Next, we wanted to understand the mechanism behind this recruitment. To do so, we did an exploratory drug panel assay with 7 different drugs that inhibited various aspects of the phagosomal maturation pathway. A result that was interesting to us was DPI, which significantly reduced APOL7C recruitment. Now, DPI acutely inhibits NADPH oxidase, which is a major enzyme complex. To explore this further, we wanted to do chronic inhibition, so we developed P22-FOX knockouts. And P22-FOX is a major subunit of the enzyme important for its functioning. In 2 successful clones, clone 11 and 19, we again saw this significant reduction in APOL7C recruitment to phagosomes, suggesting that its recruitment is NADPH oxidase dependent. Once recruited to phagosomes, we wanted to understand if APOL7C then drives escape of macromolecules. For this, we employed lucifer yellow assays. Now, lucifer yellow, shown here in green, is a membrane impermeant dye. Thus, we would expect it to remain retained within phagosomes which are membrane bound. Interestingly, we found that while APOL7C negative phagosomes did retain lucifer yellow, any APOL7C positive phagosomes saw essentially no fluorescence of lucifer yellow, which is shown visually in figure A and quantified in figure C. To further explore this, we did live cell imaging of the process and found that this leakage of lucifer yellow dye from the phagosome only occurred after APOL7C was recruited. So as the APOL7C signal increases in figure B, we then see a decrease in lucifer yellow. This suggests that APOL7C is important for triggering macromolecule escape. Finally, we wanted to understand the impact of APOL7C on cross-presentation. To do this, we employed a rather complex assay where we fed our dendritic cells ova beads. They then presented ova peptide at their cell surface. Now, specifically engineered B3Z hybridoma cells, which specifically recognize the ova peptide, and upon recognizing it, will then produce beta-galactosidase. Thus, when we introduce a CPRG substrate to the media, the beta-galactosidase will cleave it, and we can thus measure cross-presentation indirectly by measuring absorbance. Interestingly, we found that in DOX-positive dendritic cells, so cells that are going to overexpress APOL7C, there was increased cross-presenting ability, whereas in cells just expressing APOL7C at basal levels, there was a reduced ability of the cells to cross-present antigen. Together, this suggests that APOL7C is recruited to late endosomes in an NADPH oxidase-dependent manner. APOL7C then triggers the release of endosomal contents and ultimately enhances cross-presentation. I just want to say a quick thank you to my supervisor, Dr. Canton, and to all my lab mates, as well as to the O'Brien Summer Studentship Award for funding my research 2 summers in a row. Thank you.